Hello everyone. Today, Laura, Rami, and I will be presenting the 1993 Chemistry Nobel Peace Prize. To start off, we will talk about our first prize winner, Carrie Banks Mullis, who was born on December 28, 1944, in Lenoir, North Carolina. Mullis graduated with a BS in chemistry from Georgia Institute of Technology in 1966. And later obtained his Ph.D. from the University of California at Berkeley in 1973. It was in 1979 where Mullis joined a California biotechnology firm known as Cetus Corp., where he worked on his prize-winning research. Mullis developed the PCR in 1983, which would later earn him a Nobel Prize. From 1986 to 1988, Mullis worked in San Diego, California, as director of a molecular biology company known as Extronics, Inc. Apart from the Nobel Prize in Chemistry, Mullis would go on to earn several awards, which include the William Allen Memorial Award of the American Society of Human Genetics in 1990, the National Biotechnology Award in 1991, the California Scientist of the Year Award in 1992, and was also presented the honorary degree of Doctor of Science from the University of South Carolina in 1994, and finally inducted into the Inventors Hall of Fame in 1998. Mullis would go on to publish some of his writings, such as The Cosmological Significance of Time Reversal and The Unusual Origin of the Polymerase Chain Reaction. He would live his final years in Newport Beach with his wife, Nancy Cosgrove Mullis, until his death on August 7, 2019. Next, we have our second prize winner, known as Michael Smith, who was born on April 26, 1932, in Blackpool, England. In 1956, Smith obtained his Ph.D. from the University of Manchester and would later move to Canada to take on his postdoctoral fellowship at the University of British Columbia. He would spend the next 4 years working on the synthesis of biologically important organophosphates. Smith would later work as a chemist at the Fisheries Research Board of Canada Laboratory in Vancouver in 1961, where he published several papers about crabs, salmon, and marine mollusks. In 1966, Smith became a professor of biochemistry in the UBC Faculty of Medicine. From 1987 to 1995, Smith became the founding director of the UBC Biotechnology Laboratory. Smith would go on to earn several awards apart from the Nobel Prize in Chemistry, such as the Gold Medal Science Council of BC in 1984, Gardner Foundation International Award in 1986, the Award of Excellence Genetic Society of Canada in 1988, and many more. Smith would continue to work as a UBC professor of biochemistry in the Faculty of Medicine until his death on October 4, 2000, in Vancouver, British Columbia, Canada. Having won the Nobel Prize in Chemistry, both Smith and Mullis would go on to write their own Nobel lecture on their inventions. Here we have Smith's Nobel lecture titled Synthetic DNA and Biology, where he talks about the invention which allowed DNA molecules to be split into smaller segments, also known as oligonucleotide-based site-directed mutagenesis. However, Smith begins his lecture with a brief introduction where he talks about his experience and what he learned as he worked in a lab. For example, he mentions his arrival in the laboratory with his mentor, Gobind Khorana, where he was mainly involved in small molecule synthesis. Smith continues to talk about his experience and learnings which finally led to his invention. As for Mullis, his lecture was titled The Polymerase Chain Reaction, where he talks more about his personal feelings throughout his journey as he looks back at each moment in his career. He begins to talk about his opinion on the idea of a scientific creation, where he mentions some people who he looked up to, such as Richard Feynman. In doing so, Mullis tries to make the reader understand his perspective before he begins with the start of his journey. Overall, both lectures give their point of views and experiences which led up to their inventions.
Michael Smith won his prize for a paper about oligonucleotide-based site-directed mutagenesis. These are all big words, so what do they mean? An oligonucleotide is a molecule consisting of a relatively small number of nucleotides. They are short polymers of nucleic acids that can be between 13 to 25 nucleotides long. Mutagenesis is the changing of an organism's DNA that results in a genetic mutation. Mutations can include the insertion or deletion of nucleotides, as well as the switching from one base pair to another. Thus, mutagenesis is the genesis, or creation of, a mutation. Since it is site-directed, this means the researchers can pick a specific series of nucleotides, or even just one nucleotide, to mutate within a section of DNA. This is usually done on plasmids, since they are small, self-replicating, and extrachromosomal. This makes it easier to introduce foreign DNA into a cell, through a plasmid, compared to changing the DNA in a chromosome. So now we know what oligonucleotide-based site-directed mutagenesis is. The problem before Michael Smith's work was that this kind of mutagenesis could not be site-specific, and it was done through the use of radiation or chemical mutagens. Michael Smith changed our understanding of how we can edit DNA without these mutagens, and instead only with DNA polymerase, DNA ligase, restriction endonuclease, and exonuclease. Smith used the enterobacteriophage Phi-X174 to perform his experiments. This phage has circular, single-stranded DNA that can be easily manipulated and broken into oligonucleotides. The technique of mutating DNA in small fragments of this phage had previously been achieved by Clyde Hutchison and Marshall Edgell. When Hutchison and Michael Smith worked together in sequencing phage Phi-X174, they realized that it would be most efficient to use a mutant oligonucleotide primer for E. coli DNA polymerase I on a circular single-strand template. This could then be converted into a closed circular double-stranded DNA through enzymatic ligation. To create this DNA, Smith and Hutchison used a 12-nucleotide oligomer with a single nucleotide mismatch as a primer, along with the Phi-X174 DNA and modified E. coli DNA polymerase I. They focused on mutating and reverting a known nonsense mutation, AM3, involving the conversion of a TRP codon to an AMBR codon through specific mismatches. Thus, Smith helped form a new method to conduct mutagenesis artificially. This technique could take a long time to complete without multiple plasmids and primers, but the work of our second Nobel Prize winner, Carey Mullis, helped solve that problem. In order to analyze genetic information, a large amount of DNA is required. Mullis invented the technique of polymerase chain reaction, also known as PCR. Think of DNA as a paper that needs to be copied. In a copy machine, you can input the number of copies you want and have them printed on command. PCR is similar, but for DNA instead of paper documents. The original version of PCR that Mullis developed used repeated cycles of elevated temperatures to separate DNA strands, since DNA stability is decreased under high temperatures. The separated strands are then copied by a heat-stable DNA polymerase, like Taq. After repeating this cycle many times, there is an exponential growth in the amount of DNA. These steps are known as denaturation, annealing, and extension, or synthesizing. With this method, faint traces of DNA can be amplified into amounts that can be easily sequenced or quantified. Specifically, the steps occur like this. First, DNA is denatured at around 95 degrees Celsius, resulting in two single strands. We know that G and C pairs, guanine and cytosine, are held together by 3 hydrogen bonds, and A and T pairs, adenine and thymine, are held together by 2 hydrogen bonds. Well, the high heat breaks these hydrogen bonds between the opposing nucleotides, which separates the strands while preserving the nucleotide sequence. Then, the tube in which the PCR reaction is being conducted is cooled to 55 degrees, and DNA primers, nucleotides, and DNA polymerase are added to the mixture. This is when the annealing step occurs, in which the primers bind to the target DNA, ready to replicate. Once the primers and heat-stable polymerase are bound, the DNA undergoes synthesis at 72 degrees Celsius. The DNA polymerase extends the 3' end of each primer along the template strands. Finally, the original 2 strands result in 4 strands, with 2 new ones based on the template strands. When repeating this cycle 20 to 30 to 40 times, you result in an exponentially amplified DNA that would otherwise take days or months to synthesize through regular cell-conducted DNA replication. Mullis's revolutionary work relies on basic principles, like how heat affects DNA strand stability and how primers attach to promoter regions of DNA to replicate and transcribe genes. Yet, this process is very successful and can be done in a relatively short time span without the need for constantly adding new enzymes to keep the process going.
With this education behind him, Mullis started a job that would make him one of the most important scientists of the 20th century. In the beginning, he worked on developing drugs, mostly in the pharmaceutical area. Mullis made important advances to the creation of nucleic acid analogues, which are chemicals used in drugs that fight viruses and cancer. This work showed how good he was at organic chemistry and set the stage for his later successes in molecular biology. In 1983, Mullis' invention of the polymerase chain reaction was his most significant contribution, which allows researchers to amplify a DNA fragment billions of times to study genes. Two short oligonucleotides are synthesized to ensure proper bonding to the DNA segment's opposing strands. Another enzyme, DNA polymerase, may read the genetic code and link nucleotides. This creates two DNA double strands. After heating the sample, the strands separate and can be read again. After this, the technique is repeated, doubling the DNA segment copies needed. Through repeating cycles, millions of copies of the DNA segment can be obtained quickly. Mullis was involved in chemical synthesis of short sequences of DNA called oligonucleotides, which is crucial for various applications in molecular biology, such as DNA sequencing, site-directed mutagenesis, and the synthesis of DNA probes. Before his work in molecular biology, Mullis trained as a biochemist and was an expert in analytical chemistry, which is what led him to develop the PCR. Mullis' works on the PCR contributed to advancements in DNA sequencing techniques, including the Sanger sequencing method. There is no doubting the influence that Mullis has made on the scientific community. His position as one of the most brilliant minds of his generation was further joined by the fact that he made significant involvements in fields such as DNA sequencing, genetic engineering, and the polymerase chain reaction. He was then awarded the Nobel Prize in Chemistry in 1993, which was shared with Michael Smith for his contributions to the field of DNA-based chemistry. A combination of original thinking, proficiency in chemistry, and an excitement for exploring the unknown, Mullis made an obvious impression on the scientific world, and his contributions will be remembered for the generations to come. Michael Smith, another scientific researcher who had a discovery called oligonucleotide-based site-directed mutagenesis. Smith's technique opened up a route to combating certain inborn diseases through gene therapy, correcting the mutations that cause disease by rearranging an abnormal sequence of molecules in DNA sequences to the normal ones. In order to break and reform DNA chains, the synthetic segment is often added to the conventional DNA using chemicals that are considered to be standard. However, there is one thing that is not right with the synthesized oligonucleotide, and it is the fact that it contains an adenine in place of guanine. For the purpose of causing the mutation, this is done on purpose. The four nucleotides that are located below and above the adenine serve as a form of address that permits this length of DNA to align with the appropriate location on the other strand of DNA. Researchers were able to target certain genes of sections within the DNA with a great degree of precision thanks to Smith's method of the site-directed mutagenesis. By introducing targeted mutations into genes, researchers can study the function of specific genetic elements. This has been crucial in understanding gene function in both prokaryotic and eukaryotic organisms. Site-directed mutagenesis is fundamental in protein engineering, enabling the modification of amino acid sequences to alter protein properties. This has been used to enhance enzyme activity, stability, and specificity to create novel proteins with desired functions. Researchers are now able to investigate the workings of particular genetic components by introducing mutations that are specifically targeted into genes. Both prokaryotic and eukaryotic species have benefited greatly from this, and it has been essential in understanding gene function. In the field of gene therapy, site-directed mutagenesis is an essential component in the process of changing genes that are responsible for disease disorders in order to sort out genetic abnormalities. Additionally, it makes it easier to build tools for editing genes, such as CRISPR-Cas9, which offers the capability of modifying DNA in living creatures with greater precision. Scientists have utilized site-directed mutagenesis to analyze the connections between the structure and the function of protein plaques and Alzheimer's disease. They have also investigated the potential of gene therapy for cystic fibrosis and hemophilia. Furthermore, they have identified the features of protein receptors at neurotransmitter binding sites and developed analogs with unique pharmaceutical properties. Additionally, they have studied the viral proteins associated with diseases and enhanced the characteristics of industrial enzymes employed in food science. Finally, Gary Mullis and Michael Smith work together has made a big difference in the field of molecular biology. Their contributions will always be remembered in the scientific study world because of the polymerase chain reaction which Mullis invented, which has changed the way scientists copy and study DNA and is now an essential tool in labs all over the world. Smith's groundbreaking work in the oligonucleotide-based site-directed mutagenesis has given researchers the tools to accurately change DNA sequences, which makes it easier to study gene function and create new ways to treat diseases. Together, Mullis and Smith's work has changed the way we think about genetics and how to change DNA. This has opened the door to many new uses in medicine, science, and other fields, because they were dedicated to scientific research and were not afraid to question what most people thought. They moved the field forward and inspired other scientists to do the same. This is our sources we used to help create the slideshows. Hope you guys really enjoyed the show. Make sure to subscribe and like to see more content like this. Thank you.